So as the summer session draws to a close, I think it's probably helpful to try and stitch together all of the information that I've taught you in your bioinformatics lessons, that Mr. Dieff has helped you read throughout all of his audio notes, and all of the information that you've hopefully been reading on your own even without his audio notes to help you. This will give you a little bit of perspective, so as you work your way through the material in the fall for AP Seminar, hopefully it kind of gives you the big picture as you start to consider your research projects. So we started this summer by talking about the genome, and we said that the genome is all of the DNA nucleotides in any organism. That's its genome, including all of the non-coding parts and coding parts, so every single nucleotide. After that, we made sure that we reinforced the idea of what DNA does. That DNA is a storage molecule and it contains all of the genetic information that an organism is going to get. And the purpose of that genetic information is to be transcribed into RNA and then translated into protein. These regions that code for proteins, remember, we refer to as genes. And whether or not a gene is going to be used, it has a special name that we call gene expression. So remember that whether or not a gene gets used, we call gene expression. And gene expression is influenced by a lot of things, including environment and hormones. And I do believe it was our second, maybe our first lesson, where we talked about gene expression being very different in prokaryotes and eukaryotes. But really, it's just how often a gene is transcribed and translated. And we call that gene expression. Then we talked, I don't know if we explicitly talked about specifically what a protein does, but I know in our last lesson we said that what proteins do are very heavily influenced by their shape. So a protein is nothing but a sequence of amino acids. And what amino acids a protein contains and their R groups, those side chains, the variable groups, are going to influence the function of that particular protein. This is where I can add in a little bit from hopefully what you've read in your readings about genome-wide association studies, how scientists are using these GWAS studies to determine whether a variation in a genome is linked with a particular trait or disease. Now, I just used a word there that is actually a pretty important word that we'll talk about for our next slide. Differences in DNA sequences being variations. So we already have a kind of an understanding that every organism's genome is different than every other organism's genome, which means your genome is different than my genome, and our genomes are different than that of a dog, a cat, or a cow. These differences between a species, so between, say, human and cow, there are a greater number of variations and differences than there are between human to human. These variations are simply just changes in genes, changes in DNA sequences. And these differences, those variations, as far as humans go, variations are typically just the difference between inheriting different alleles. Remember, an allele is a different version of a gene. So this kind of takes us back to that very last section in honors biology where we used one of our examples of different alleles. We talked about blood types. So all humans have blood. All humans have proteins that control what's on the surface of their blood, but the allele or the version of the gene that you get to make that protein can cause different shapes of protein. So we end up with different blood types, type A, type B, type AB, and type O. The phenotypes are different because the allele is different. These are just simple variations in a gene sequence. Simple single nucleotide changes, sometimes single nucleotide, sometimes multiple nucleotide changes, that cause a change in the protein, which causes a change in appearance of that protein, or it's not necessarily its function, it still does the same thing, but it does so differently. As we started to really consider what these differences looked like, or how we find them, this is where we looked at genomics and our databases that contain entire genome sequences, because we analyze those genome sequences for genes. We look at the particular amino acid sequence that those genes code for the protein that they make, and we can use those in a variety of different ways. A couple of the things that I asked you to look at were differences between species, because those differences allow us to examine biodiversity, allow us to establish taxonomy, and they allow us to establish evolutionary relationships between organisms. So that's one use of all this data. And there are different kinds of variations, though, that we didn't talk about together in bioinformatics, but you did have the opportunity to discuss or to think about in your readings. Those differences are not differences in DNA sequences, they're differences on DNA sequences. And those differences on DNA sequences we call epigenetics. Where the prefix epi means on or upon, and it refers to chemicals that can attach themselves to DNA in a cell, and those chemicals influence gene expression. So those chemicals attaching themselves to a cell can turn genes on or turn genes off. It can turn gene expression levels up, or it can turn them down. Epigenetics and changes to the epigenome are in fact heritable, and they change gene expression without altering the DNA sequence. So it kind of takes everything that we've talked about and makes it a bit more complicated. That it's not simply the difference between what your nucleotides say, but it's also the difference between what's attached to your nucleotides. Changes in epigenetics can really cause some significant problems, can cause diseases, but changes in epigenetics are also helping us treat diseases. So it's a whole field within genetics that's pretty interesting, because it means that it's not just as simple as nucleotides or base pairs. That's kind of really all of the biology that we've done this summer. We've talked a lot about the genome and how we analyze the genome, and some of what I've asked you to do, actually a lot of what I've asked you to do, isn't exclusively biology. It's bioinformatics, how we take the biology that we know to be true and we use computer science to help us analyze what we know to be true, to help us gain further knowledge or understanding of how organisms work, how organisms are related, and how even within a single organism, so say a human itself, what each of those genes is going to do and what that protein product does. And that's a pretty big thing to think about. But we're going to ask you to actually think a little, a lot, more deeply about it, and think about an even bigger picture. So the question I have to ask then is, we've looked at genomes and we've looked at variations and we've talked about alleles. Are variations in a genome good, or are variations in a genome bad? One of the questions Mr. DF asked you guys to begin to grapple with in one of his most recent emails where he sent his audio notes, he asked you whether we should all be the same or we should all be different. Is it good to be different, or is it good to be the same? Variations can cause disease. In your reading of Francis Collins, you encountered Huntington's disease, BRCA1, which predisposes an individual for breast cancer. I do believe Francis Collins in that same text mentions APOE. That would be the Alzheimer's, quote unquote, Alzheimer's gene. He, I think in that same text, says that the Alzheimer's gene isn't as simple as, it's not as simple as inheriting APOE, but it's also correlated with head injury. You also read about cystic fibrosis in Francis Collins, as well as Marfan syndrome. I know that was one that was mentioned in the Collins text as well. So these variations between humans, they can cause some pretty drastic, significant diseases. So it seems like in this way that variation would be pretty bad. But he also says in that same text, Francis Collins says that you could have a strengthened immune response because of genes that are inherited or because of variations in genes. Those variations can cause a strengthened immune response to sickle cell or to malaria. And that's also pretty important. People who live in regions where those are common diseases, having that variation would be a very positive thing. Francis Collins, as well as a few other biologists, also point out that variations cause differences in phenotype. Now, that's always true. That's a pretty simple statement. Variations in a gene cause differences in phenotype, or can cause differences in phenotype, if the substitution that's made, if the change that's made influences the shape of the protein. Is that always good or bad? That's the question I have to ask you. Is having type A blood any better than having type B? Is being type AB any better than being A or B? Sometimes a change in phenotype is simply a change in phenotype. We assign the words good, bad, better, detrimental, negative, positive. All of these are words that imply some kind of value judgment. And your value judgment is entirely dependent upon your own personal value system. So differences in phenotype, variations cause differences in phenotype. I think one of the examples Mr. DF posted on Google Plus is, would you edit your child's genome for IQ? Is IQ, a difference in phenotype, a difference in IQ, is that good or bad? Is it always good to be high IQ? Does that mean that you've made a judgment about someone who is not high IQ? That you have devalued them as a human person because they don't have the native intelligence, the intellectual capacity that you find valuable? So, are these variations good or are they bad? The consequences of bioinformatics as it applies to humans kind of leads us naturally in that direction. Bioinformatics, as we apply it to human beings, we look at diversity and variation. Is diversity a good thing or a bad thing? What causes diversity among humans? What causes variations among humans? The eugenics movement. You should have early in the semester read a lot about eugenics. And remember, EU, that means good, favorable, positive. So we're using genetics or manipulating genetics to try and maximize favorable traits. Who determines what favorable traits are, is the question that leads to. Bioinformatics, as it applies to humans, allows us to do those GWAS studies I mentioned earlier and determine linked traits to a particular variation in the genome. But then we have to ask ourselves is how accurate are those GWAS studies? Hopefully you've been reading or learning some statistics as well as learning some computer programming along the way this summer, and you recognize that those GWAS studies rely upon statistics and computer programming, and if someone hasn't constructed their program properly or hasn't done their statistics properly, you get a lot of false positives in GWAS studies. So that's something to keep in mind. Bioinformatics as it applies to humans, and biobanking DNA. I think one of Mr. DF's most recent audio notes includes whether or not they should be able to biobank DNA for criminals, as well as anyone whose DNA has been collected by law enforcement for the purposes of an investigation. Do they keep those things on file, and do you have a right to your genome? I think one of the most recent Google Plus posts that I read was about hackers attempting to hack medical systems to access people's DNA. What does that mean? What are the consequences? Is your DNA yours? Do you own it? Do you own the information that it contains? These are all pretty big questions that aren't necessarily biology. They certainly involve biology, but they're not necessarily biology, which means that we have to have a very strong foundation in ethics, morality, political science. It's pretty big questions. Another thing to consider for bioinformatics is direct to consumer genetic testing. There's a company called 23andMe that you can, submit your DNA to it and they'll test you. I think all they're allowed to test you for right now, because of a cease and desist order from the FDA, all they're allowed to test you for is your, what do you call it? Country of origin. So inheritance, ancestry. So it's for ancestry purposes, but they used to test you for diseases. And unfortunately, they weren't providing people with enough counseling when they gave them those results. And it had some pretty negative effects. 23andMe is not the only direct-to-consumer genetic testing out there. There's a couple of other companies like KnowMe, like KNOW Me. They do the same thing. They attempt to test people for particular diseases or traits is usually what they're looking for. Bioinformatics applied to humans also allows us to do personalized medicine. Personalized medicine is very likely the direction that medicine is going to start going as whole genome sequencing becomes less expensive and more accessible to the consumer. How to treat you, recognizing what genes are in your immune system. So how to maximize your own immune system and minimize the damage to your immune system by designing the perfect complement of medicine to give you based on your genes. I know that Vanderbilt is pushing their personalized medicine pretty hard. Like, I've seen quite a few commercials for their own medicine or their personalized medicine because they're using it in cancer treatment and having some very positive results. Some of the other things we've asked you to think about this summer, gene editing and CRISPR-Cas9. So CRISPR-Cas9, remember, is just a technique for gene editing where they can go through and change what the genome says. Should we be changing what the genome says? Are we trying to get rid of variation? All variation? Are we trying to just get rid of variation that we've determined is negative? A lot of ethical questions that I'm sure Mr. Sierra adequately prepared you guys to try and start to wrestle with. And then, of course, there's always the off-target effects of gene editing. All of these things that we've looked at here, the consequences of bioinformatics, are simply, and it's really not a comprehensive list, but they are simply the result of the bioinformatics movement. How we can take a huge amount of data, analyze that data, and after analyzing that data, what do we use the results for? I've been trying to show you guys over the course of the summer how to analyze the data, because that's likely where you're going to be the most successful in your research projects. You'll be working with qualified scientists who will help you analyze gene data. But I don't think that that's all Mr. DF and I want you to do. What we're really interested in is saying, we've prepared you to become these scientists who work in bioinformatics. We've prepared you to analyze a huge data set, but we've prepared you to do so with the understanding of the implications of the results of that analysis. That if you analyze these things, what could we do with that data? What should we do with that data? And how do we establish these rules? That's kind of been our goal this summer, is to get you to think about those things. And hopefully you've gained a ton of knowledge and a ton of information. And if that information seems overwhelming, or if it seems like there's a lot of it, that's perfectly okay. That's normal, and that's the way it should be. Truthfully, the reason that it's overwhelming for you is because it's overwhelming for all of the scientists who are out there asking the exact same questions. So that's kind of our goal here. That's what we're looking for, and that's what we're going to prepare you to do as you work your way into AP Seminar and on your capstone project. So I hope all of this kind of helped stitch together some of those vocabulary words, give you a little bit of perspective. I'm going to continue to try and see if I can't develop a picture or flow chart to make it all fit together. As I was writing down my notes for this particular lesson, I wasn't sure. I didn't have the best picture yet, and I don't think I have one that's satisfactory because there's so many huge ideas. I'm going to keep thinking on it, and when I do, I'll post it for you guys. All right, I hope you guys have learned a lot. I hope you guys have stayed caught up on your readings. If you're not, there's a little bit of time to catch up. And I will see you, I think, at U of M Dearborn. I do believe we have a date scheduled. I just have to check what it is. Good luck. See you then.